Thank you. Again, I want to thank the organizers for having me here. And I'll take you through the brain in about 50 minutes, I guess. I'll first talk about exercise and neurotransmission and how exercise influences neurotransmission. Then talk about the energy the brain needs, not only during exercise, but in general. And then, of course, I'll go into the fact of to see if exercise is really good to the brain, because we will be talking about neurogenesis, the new formation of neurons. And then I'll end with a specific aspect: is exercise good for the brain when you exercise in a polluted environment? So there are several neurotransmitter systems, and I just give you a few examples, such as the Neurotransmitter System, the Dopaminergic System, and the Serotonergic System. And as you can see, the cell bodies are located low in the brain, near the brainstem, and they send signals around all over the brain. For instance, here for the Dopaminergic System, the nigral striatum system, from the substantia nigra to the striatum, is very important in Parkinson's disease. If about 90% of the dopamine, which is located here in the cell bodies, is exhausted, then the symptoms of Parkinson's disease start emerging. While the ventral tegmental area, this brain area, is, you can call that the pleasure center of the brain. If you put some stimuli in that brain area, specific, for instance, in rats, then they will continue running until exhaustion, and even beyond the normal running capabilities. And of course, to make it simple, the higher location of your cell bodies or neurons in the brain, the more it's your active brain that is working, and the lower in the brain, let's say, it's more your automatic pilot that is working. So I already showed you this morning that I was very interested to look what was happening in the extracellular space, and that we use the microdialysis technique for this. And microdialysis is a sampling technique in which you harvest substances from extracellular space, but it can be done in any body or any tissue, actually. It uses a dialysis principle, where there's a concentration gradient at the tip of that little needle that you implant. There will be a membrane which collects substances from the extracellular space that are not in the perfusion fluid. And you have a very low perfusion fluid, so you can harvest during 20 minutes, let's say 20 microliters, or if you have a double speed, then you do 40 microliters, which you